For over 50 years, the Nobel Conference at Gustavus Adolphus College has hosted preeminent scientists, theologians, and ethicists to discuss questions at the intersection of science and society. From the newest results in physics, chemistry, and biology to the newest fields of multidisciplinary study, scientists at the Nobel Conference have examined the universe at its largest and smallest scales, explored the oceans, described new materials, and investigated human and animal behavior. Conference speakers have debated the mechanisms of aging as well as the science and economics of food. Often, speakers have given us a glimpse of the next defining questions and how they might be answered. Throughout all of the conversations, ethicists and theologians have grounded the science in a human dimension. This year's conference focuses on addiction, a uniquely human condition. The 51st Nobel Conference, Addiction, Exploring the Science and Experience of an Equal Opportunity Condition, brings together experts in medicine, neuroscience, sociology, and philosophy to explore the science and experience of addiction. The conference explores a range of questions. What does it mean to be addicted? Is it a brain condition, a psychological disorder, a sociological problem? The answers to these questions raise a very important one. What treatment options are available? We hope you enjoy this year's presentation by Denise Kandel, who is co-presenting with her husband, Nobel laureate Dr. Eric Kandel. Their presentation is entitled Molecular Basis for the Gateway Hypothesis. Denise B. Kandel is professor of socio-medical sciences at the Department of Psychiatry at Columbia University School of Public Health. Dr. Kandel is known for her pioneering studies on the developmental phase of drug use and her identification of the specific risk factors for adolescent initiation into each major stage. This work provided the basis for the gateway hypothesis. Her current work focuses on smoking and nicotine dependence in adolescents, and she has begun looking at the molecular basis of the gateway hypothesis with Dr. Eric Kandel and Dr. Amir Levine from the Center for Neurobiology and Behavior.
Well, thank you very much for this wonderful introduction. I don't know what else to say. The only thing I want to say is if you start with cigarettes, you don't necessarily become a heroin addict. Anyway, I want to, before I present my talk, I want to thank very much the college and the wonderful reception they've given us, and I want to highlight in particular Scott, Burr. We had many emails before all this got accomplished, and once we've been here, I've had two wonderful students taking care of me, Anna Hollander and Clayton Mickelson. And I want to thank them very much. They made us feel very much at home, and I feel very warm toward Gustavus University. So let me start, and in fact, I will talk about the gateway hypothesis, try to explain what it is and what some of the underlying factors are. And what it is the gateway hypothesis describes a developmental process of involvement in drugs in which certain drugs serve as a gateway for the use of other substances in a specific temporal sequence. So in a sense, what we want to say is that once you use a particular drug, your risk of progressing to another drug increases. But as I said, it doesn't mean that because you smoke a cigarette, you're going to become a cocaine or a heroin addict. And in my talk today, I will try to cover different perspectives of the hypothesis, but not everything, obviously. Ranging from epidemiology to biology, and as far as the biology is concerned, my husband will take over. And so I will present epidemiological data that describe the sequence that is typical in Western societies. My husband will discuss findings of the molecular mechanism that underlies the hypothesis from work done in collaboration with his laboratory. I will describe briefly the implications of animal results for human populations. I will also discuss the implications of the gateway hypothesis for understanding broad trends in drug use in the population among different birth cohorts, and I will conclude with a very short discussion of e-cigarettes. I don't think we can ignore e-cigarettes today. So let's start with the epidemiology. There are two large monitoring efforts going on in the United States at present, in which the government tries to gain some knowledge about the use of different drugs in the population. So one set of studies, and these studies are done on an annual basis using the same methodology so you can follow trends over time. One is a national household study of health, which study individuals 12 years and older that are interviewed in their homes. And the second study is Monitoring the Future, which is an annual study of 8th, 10th, and 12th graders throughout the United States. So drawing from the National Household Survey on Drug Use and Health, I focus here on individuals who are aged 18 to 34, which is a part of the population that is most heavily involved in drugs. And these are very simple data showing you the proportion of individuals who use different drugs. And so the most prevalent drug is alcohol. Almost everybody has drunk. Among these people, 18 to 34, 2/3 have smoked. More than half have used marijuana. Very few have used heroin. You don't find heroin users in the general population. And if you look at the mean age of onset, you see the first drug that's used is cigarettes on average, about 16 years old, then alcohol and marijuana, and the last drugs is heroin. So you can use these data about mean age of onset without having longitudinal data to try to understand what is the sequence in which people use drugs. So this next chart shows you the proportion of people who have used marijuana, the proportion among these people who started smoking and drinking first. You ask them, how old were you when you did this very thing? So you say 63%. Who started smoking, drinking, and using marijuana at the same age. Started marijuana first, 13%. Never smoked, nor drank, no one. So you see the small proportion who started with marijuana, but the majority of people started smoking and drinking. If you focus on smoking per se, you see that 40% use smoking first, anyway, but 14% never smoked. If you look at cocaine, the sequence is even stronger. So you looked at the relationship between smoking and cocaine, and you see 89% have smoked before they use cocaine, and only 3% never smoked. Same thing for marijuana. Most cocaine users, 89% have used marijuana, and 2% have never used marijuana, and only 2% started cocaine first. So you find the sequence in different countries throughout the world. I did, for instance, a long time ago, a study in France and in Israel, and you found exactly the same sequence, even though the prevalence of the use of different drugs varies in the different countries, such as, for instance, in Israel, many fewer people drink than they do in France. And so this cartoon provides visually depicts the pathways in which people start using alcohol and cigarettes, and then if they use one, they then proceed to use the other. And if they proceed further, it's marijuana, then cocaine, and heroin, and there are different pathways and deviation because not 100% of the population, there's something like that. And the thickness of the bars indicates approximately the proportion of people who go into the different pathways. So the term gateway hypothesis was introduced to emphasize that certain drugs serve as gateway for the use of other substances in a specific temporal sequence. Now, I want to emphasize that patterns are changing among young people, and everybody has heard about the legalization of marijuana in many states, the increase in the use of marijuana. So if you focus upon the behavior of 12th graders for monitoring the future, the second study that I mentioned to you, you see that the proportion who've ever smoked, 38%, is now lower than the proportion who use marijuana. And the proportion of alcohol is the highest, and very few kids have used cocaine. And you see these stands have contrasted the data for the seniors from the people 18 to 34. And so you can see very well what the kind of historical changes that are taking place now. So this, of course, is going to influence the sequence. So if you look at the 12th graders and you say, well, what proportion have smoked or drank alcohol before using marijuana? You still find it's a majority. There are only 13% who started using marijuana first, have smoked or drank And a high proportion started all three at the same age, and only 3% never drank or smoked. If you look about cigarettes, this is really where you see the strongest change. You can see 37% of the marijuana users have never smoked. Only 26% smoked first. So if you compare what happened among people 18 to 34 and the 12th graders, this really summarizes the contrast very greatly, in which you see the proportion who never smoked amongst 12th graders is much greater than the proportion among 18 and 34-year-olds. But these are changes that have taken place, very, very recently. And if you look at the sequence between smoking and cocaine, it's changed, but still, even among the 12th graders, now you found almost 70% have smoked before they've used cocaine. So the gateway hypothesis provides an important framework for understanding the risk factors and consequences of different forms of drug involvement, and the link between nicotine and cocaine is especially strong. But there are limitations to the epidemiological approach, and surprisingly little progress has been made in addressing the two fundamental questions that derive from the observation that the use of one class of drug is followed by the use of another. Does the use of the first drug cause the use of the second drug? And what mechanisms underlie drug use progression? So, biology can be experimental, but sociology is more of an observational science. And so, although the epidemiological data have established that there is a sequence and there's an association between them, these studies cannot establish a causal progression, nor can they identify the underlying cellular and molecular mechanism that could contribute to the gateway sequence of drug use. And in the question period, I'd be glad to answer why epidemiological studies can do this. So a sequence might be due to a common genetic vulnerability for substance abuse and the relative availability of different drugs. You can say, well, certain individuals have a genetic tendency toward using drugs, so it doesn't matter which drug, they're going to use the ones that are readily available. But the other other interpretation is that certain drugs may lead to long-lasting changes in the brain that enhance the sensitivity to other drugs and increase the vulnerability for abuse and dependence. And I will say the two interpretations are not mutually exclusive. I think that's very important. So in order to try to understand more deeply what's going on, we're championing an approach called translational epidemiology, in which you go from epidemiology to molecular biology, and the reverse. You can go from molecular biology to epidemiology. What does that mean? That means that you take an observation that you've observed in human population, epidemiology, and you try to develop a model in animals in which you can then study the underlying biological mechanism. And so this is what we've done. So we looked at the association between nicotine and cocaine, and so we bridged the epidemiology and drug abuse and molecular biology by developing a mouse model of this sequence. So we could explore the behavioral, physiological, and molecular mechanism underlying this gateway sequence. But the problem in doing this as an epidemiologist is to find a biologist who's going to want to collaborate with me. And over 2 or 3 years, I really had a very hard time. And then one day I realized, before we go into this, I want to emphasize why animal models are very important, because they provide a rigorous test of the sequence. You can look at the behavior in relation to well-defined prior experience with specific drugs, independently of social or legal constraints regulating and defining drug use. And it's a reductionist approach to which some people in this audience, I know, are going to object strenuously. But what is very important is that alternate specifications of the sequential order of drug presentation can be defined. So you can prime an animal with nicotine, then expose it to cocaine, see what it does. You do the reverse. You prime it with cocaine, then expose it to nicotine and see what it does. You see whether the order makes a difference. So it becomes the only experimental determinant of outcome. So it's very hard. I was looking for a collaborator, and then one day I found him. In collaboration with Eric and Amir Levine, we developed a biological model of the gateway sequence between nicotine and cocaine, and we applied it to mice, and Eric will explain to you what he found at the molecular level. I'm going to come back.
Denise used to tell me and other people that she didn't marry me for my looks, she married me for my brains. Now I realize she married me primarily for my bow ties. The early work of Winkler and more recent work of Eric Nestler established that addiction is a form of learning. The molecular pathways involved in addiction share some of the same molecular logic and some of the same molecular steps encountered in long-term memory storage that we considered this morning. And the transcription regulator CREB is a key switch from short-term to long-term memory, and the immediate early genes, such as FOSB, lead to the growth of new synaptic connections. So I discussed this with you earlier today. So cocaine, for example, activates the dopaminergic system, which increases the level of cyclic AMP, activates the cyclic AMP-dependent protein kinase, translocates to the nucleus, phosphorylates CREB, turns on FOSB, giving rise to the growth of new synaptic connections. In 2001, my colleagues and I collaborated with Dimitri Tanos, and we found to our surprise that in Aplysia, this now holds true for the mouse as well, the CREB-mediated cascade of gene expression and growth necessary for memory storage resembles cellular differentiation and being initiated by remodeling of chromatin structure resulting from histone acetylation. This was the first demonstration that learning has similarities to developmental processes. Chromatin is a combination of DNA and protein spools. I'll show you cartoons of that in a second, called nucleosomes. The DNA is wrapped around these nucleosomes in the nucleus. Nucleosome spools consist of an octam of histones containing 2 copies each of 4 different types, H1, H2, H3, H4. So this is chromatin. It consists of these protein spools and the DNA wrapped around them. The nucleosomes contain, as I said before, 2 copies of each of 4 different histones, H1 to H4, and a cascade of gene expression is initiated by acetylation and neutralization of the positive charged histone tails by the CREB binding protein, CBP, which opens up the promoter or enhancer region, allows transcription of delta FOSB to occur. So this is the way it occurs. You have CREB1 sitting on an enhancer region or promoter region. I'm using them interchangeably. When serotonin or dopamine comes along, they activate cyclic AMP-dependent protein kinase. It phosphorylates CREB, which allows it to recruit the CREB binding protein. The CREB binding protein acetylates particular lysine residue in H4. That opens up the promoter, allows it to recruit the Tatabox binding protein and polymerase II, and allows gene transcription to occur. So this has turned out to be a relatively universal mechanism for controlling gene expression. In 2006, Amir Levine and the Nestler lab and I independently found that what holds for learning also holds for cocaine. So it's likely that addiction also involves chromatin changes. These early studies indicated. So cocaine induces gene expression of FOSB, a CREB-mediated target. It recruits the CREB binding protein to the FOSB enhancer and increases histone acetylation. So FOSB is induced by cocaine compared to control. It recruits the CREB binding protein and leads to histone 4 acetylation on the FOSB enhancer. This put us in a position to respond to Denise's suggestion that we try to test the gateway hypothesis and to see what molecular biology can contribute. So what we've tried to explore together in this collaboration is if the sequence is biologically causal, how does this causality work? That is, if you give nicotine before cocaine, does it make any difference in giving cocaine before nicotine? And if so, how does that difference come about? Does the use of specific gateway drugs such as nicotine enhance the sensitivity of another drug such as cocaine? What are the underlying mechanisms? And are the same molecular steps involved as have been found to play a role in addiction? For example, is priming due to remodeling of chromatin structure resulting from histone acetylation? And we've been able to address these several questions. So let me first begin. We looked at level of analysis at several different levels. We looked at the behavior, the neurophysiology, and molecular processes. And I should try to tell you, to expand the thinking of modern neuroscientists, is that behavioral analysis is absolutely essential to modern neuroscience analysis. You can't do meaningful analysis of behavior unless your behavior is as rigorously done as the molecular biology. Thank you very much, Denise. We first used behavioral sequence administration to study whether the sequence mattered. Does it matter whether you give nicotine before cocaine or cocaine before nicotine? We used locomotor sensitization as well as place preference. I'm only going to show you locomotor sensitization. Locomotor sensitization is essentially you give a mouse something and you see whether it affects their speed of running. We did this in two ways. We gave nicotine first and then cocaine, and this is just the control, or we gave cocaine first and then nicotine. We found that if you give nicotine alone, it does practically nothing compared to control. If you give cocaine alone, it does something, but it's not spectacular. But if you give nicotine, which does practically nothing before cocaine, you get a dramatic enhancement of the effects of cocaine. This simply shows you nicotine and cocaine compared to nicotine alone, or giving one before the other, giving cocaine first and then nicotine. When you give cocaine before nicotine, has no effect whatsoever. If you give nicotine before cocaine, have a dramatic effect. We next use this paradigm to examine three cell biological molecular markers: the priming effect of nicotine or cocaine, and cocaine and nicotine on synaptic plasticity, transcription of force B, and recruitment of histone acetylation. We focused on the striatum, which is where most drugs of abuse exert their addictive action. The first thing is we looked in the nucleus accumbens, a structure that inhibits dopaminergic cells of the VTA. We found that a very interesting effect happens here. That nicotine enhances LTP changes induced by cocaine, but cocaine has no effect on the action of nicotine. So what is interesting about this is LTP is a synaptic facilitation. But in the nucleus accumbens, it's primarily inhibitory on the VTA, the ventral tegmental area. So it inhibits the dopaminergic neurons. So the more you reduce LTP, the more you enhance the action of the dopaminergic neurons, the more you enhance the addictive process. So this is the control by itself. Chronic nicotine shows slight enhancement. Acute cocaine shows a depression, but chronic nicotine, which practically does nothing by itself over control, when you give that before cocaine, you get a dramatic depression, enhancement of dopamine action, and therefore increased likelihood for addiction. The expression of force B is induced by cocaine is enhanced by treatment of nicotine, but force B expression induced by nicotine is not enhanced by cocaine treatment. So again, you see this big enhancement when you give nicotine before cocaine. If you give cocaine before nicotine, you don't get an increase. What are the molecular mechanisms of this nicotine priming? We found that the increase in expression of force B gene induced by cocaine following nicotine pretreatment is mediated by a change in chromatin structure, an epigenetic change in the force B gene region. There's an increased acetylation of histone nucleoproteins that packages DNA into chromatin in the striatum, where the nucleus accumbens is located. And this is really quite fascinating, because it really shows you, on a molecular level, why nicotine before cocaine is so much more effective than cocaine before nicotine. So you look at cocaine alone. Cocaine comes along, it activates cyclic AMP-dependent protein kinase, activates CREB1, good beginning, recruits the CREB binding protein, which acetylates a lysine residue, so you're beginning to see chromatin structural changes, and it opens up the force B enhancer so the Tatabox binding protein can be recruited and PAL2 can be recruited. So far it really looks like it's working very well. But there is a histone deacetylase that inhibits the action of the histone acetylase. And within a very short period of time, the action of cocaine is short-circuited because all of this action of the histone acetylase is eliminated by the inhibition through the histone deacetylase. So what does nicotine do in order to improve the situation? What nicotine does is to inhibit the deacetylase. So now when cocaine comes along, it not only activates genes, but allows them to be activated for long periods of time. It dramatically enhances the time course of it. So now when cocaine comes along, activates pKa, it recruits the Krebs binding protein, reduces histone acetylation, gets transcription going. It can continue for a long period of time because the histone deacetylase, which normally removes the effects of acetylation, is inhibited. So, in summary, there are three differences between cocaine and nicotine. Cocaine acetylates the phosphate promoter site only on histone H4, whereas nicotine acetylates both H3 and H4 to the degree that cocaine cannot enhance it much further. Although cocaine acetylates H4 as much as nicotine does, it does so only locally at the phosphate promoter, while cocaine has global acetylation. By contrast, nicotine increases global acetylation at H3 and H4. So together, they're much, much more effective because nicotine has these special actions, particularly in relationship to cocaine. Moreover, this effect is clearly unidirectional. Nicotine acts as a gateway drug and exerts a priming effect on cocaine through increased histone acetylation, which increases phosphate expression in response to cocaine. Reversing the order has no impact whatsoever. So one of the interesting things that Denise found, that we also found independently, is nicotine enhances the effects of cocaine, but only if nicotine is on board at the time that you take cocaine. If you interrupt it by just a few days, nicotine has no priming effect. So nicotine has to be there when cocaine is present. And this is essentially what this shows, that nicotine has to be there, otherwise there's no difference between them. So to conclude, a biological explanation of the gateway effect, the nicotine primary effect is unidirectional. Cocaine does not prime nicotine. A new molecular epidemiology of drug use, beginning a molecular insight into the gateway effect, global acetylation by nicotine. Nicotine achieves global acetylation by inhibiting the deacetylase. This may explain the cognitive enhancing effect experienced with smoking. And priming effects of nicotine require concomitant administration of cocaine. They have to be on board at the same time. And implications for epidemiology, biology in the service of epidemiology. This is the first time we ever worked together, number one, I mean, in a non-social way, in an academic way. And it really was a very enlightening experience for both of us. Awkward in the first few moments, but then became extremely enjoyable. And I think this is a very powerful approach, bringing disciplines together in this way. And Denise is going to continue.
So the mice experiment were guided and started on the basis of the epidemiological finding. But I think the important point is that the findings in the mice research can also suggest hypotheses and ideas to be tested in humans. And Eric just pointed out that in order for the priming of nicotine to occur, it has to, the cocaine has to be taken at the same time. The animal had to be exposed to cocaine at the same time as nicotine. So I went back to a study that I conducted a long time ago in which I had tracked the drug behavior of people and asked them to reconstruct on a monthly basis what they had consumed. And I found that in testing the ideas that the priming effect of nicotine requires concurrent administration of cocaine, I had to really show two things. One is, do in fact people who start cocaine, are they in fact actively smoking at the time that they start? And does it make a difference in terms of the effect of cocaine, whether or not you were smoking at the time that you started? And so in following this relatively small sample of cocaine users, I found that in fact 75% were using the months, they were smoking the months before, the months prior to the month that they started cocaine, and only 7% started cocaine before smoking. I went also to a national data set, not the two that I mentioned before, and I found that in fact, 87% were smoking in the year in which they started cocaine, and 4% only started cocaine prior to smoking. So then the issue is, if you look at, for instance, the persistence of cocaine use, is there going to be a difference between people who are actively smoking at the time that they started cocaine and those who were not? It's actually hard to test this hypothesis because the majority of people were smoking at the time that they started cocaine, so there are very few who weren't. And when you do this in this national data set, you looked at the people who started cocaine a year earlier, at least, and you said, are they currently using cocaine? And you find that when they were actively smoking, 7.5% compared to 4.5% were still using cocaine compared to those who were not smoking at the time of cocaine onset. So that's a small example of how the biology influences epidemiology. But I also want to now move to another level of consideration and propose that if you understand the drug behavior of birth cohorts in adolescence, you can really predict what the drug behaviors of birth cohorts are going to be when they are adults. And so the hypothesis is that at the population level, the drug behavior of adults from different birth cohorts will be shaped by patterns of drug use, I'm repeating what I said, that they experience in adolescence. So that in different historical periods, the levels of cocaine use in the adult population will vary as a function of levels of smoking that these cohorts live through in adolescence. And this can be useful in terms of social policy and trying to plan for the future. So we tested this hypothesis. So optimally, you need to have longitudinal data available from adolescence to adulthood in different birth cohorts, but those data don't exist. So what you can do is you can look at repeated cross-sectional surveys that are based on national representative samples of the population, because at any one point in time, members of a particular birth cohort are assumed to be equivalent to other members of the same birth cohort. So you can follow people who were born, in the year 1990, and then follow them over time in this consecutive cross-sectional surveys. And that's what I did by using data from Monitoring the Future. I mentioned the study of high school students. Ooh, there's a spider. And I couldn't follow these people for 20 years, but I followed the drug behavior of 12th graders as a function of level of cigarette use they experienced 4 years later in 8th grade in 18 birth cohorts from 1991 to 2002, 2007. And this interval covered periods of increase, changes in the prevalence of smoking. There were low prevalence, higher prevalence, and then a great decline in the prevalence. So there were 3 contrasting high school cohorts. And in 1991, you see 44% of the seniors said they ever smoked. They were not smokers, actually, they were the 8th graders, I'm sorry. Then in 1996, 49%, and in 2008, tremendous decrease. We know that smoking has really decreased tremendously, only 20.5%. So I looked at the change in the prevalence of smoking and correlated that with change in the prevalence of cocaine use 4 years later. And what you see is that, so from 1991 to 1996, there's a period of increase in prevalence of 8th grade smoking. There was a 12% increase. And in 12th grade, 4 years later, when you looked at these cohorts, there was a 43% increase at the level of all, kind of population level of the 12th grader. And correlatively, when you looked from 1996 to 2008, when there was a very sharp decline in the rate of smoking, which was 59% among the 8th graders, 4 years later, there was a 43% decline in the rates of cocaine. So you don't need to know the behavior of individuals. You can really almost predict what's going to happen at the population level when you know what took place in adolescence. So now let me just say a few words about e-cigarettes. I'm sure everybody is interested in e-cigarettes. I'm terribly interested in e-cigarettes. It's a real phenomenon. And I think that the mass data that we presented really help inform the current debate about the potential benefits or dangers of e-cigarettes as a tool for reducing cigarette smoking and its health burden in the population. So people in favor of e-cigarettes emphasize the advantages of the fact that it's so-called clean product. Actually, we found out more and more it's not so clean, without the morbidity associated with combustible tobacco. And the morbidity is generally focused on the cardiovascular and respiratory system. I've never read anything about potential effects on the brain except about 5 months ago when CDC released the data that I'm going to show you about the changes in the prevalence of e-cigarettes among young people. So now the users include not only the people who are chronic smokers who want to stop smoking, but they include adolescent young adults, among whom the use is increasing rapidly, exponentially, until they are not the chronic middle-aged smoker that's considered to be the ideal consumer that had been envisaged. And even for the chronic smoker, it's not clear that e-cigarettes are better cessation strategies than other strategies such as the patch or gum. So the use of e-cigarettes among high school students has increased 3 times in a 1-year period. From 2013 to 2014, you can see 4.5% said that they were current means last month, using e-cigarettes among seniors, high school students, in 2013 versus 13.4% in 2014. I mean, this is an extraordinary increase. And now, if you look, for instance, at the relative use of cigarettes compared to marijuana, look at this. 13%, this is in the last 30 days, monitoring the futures, the high school study I mentioned, versus 21% of marijuana. And in fact, e-cigarettes are much more prevalent than combustible cigarettes. 17%, if you look at as the seniors use either one, it's 23% higher than marijuana. But what's also interesting is that if you look at the people who use combustible cigarettes, these are regular cigarettes, or the people who use e-cigarettes, if you use one product, you're likely to use another one. So more than half of the young seniors who use combustible cigarettes also use e-cigarettes, and 42% of those who use e-cigarettes also use combustible cigarettes. Now, the thing that we don't really know is what is the order. Do e-cigarettes precede the use of combustible cigarettes and put people at greater risk of using combustible tobacco? Or is it the case that people start with combustible cigarettes and then are more likely to start e-cigarettes? There's one study that appeared about a month ago in JAMA, the Journal of American Medical Association, suggests that in a number of cases, e-cigarettes may precede the use of combustible cigarettes. But we really don't have very good data. So, as I said, the nefarious consequences on brain functioning of the kind that we reported are not considered in the debate around e-cigarettes. You see that these data were released by the CDC in April. It's in that report that Thomas Frieden, who's the head of the CDC, said, we have to be concerned about the effect on the brain of adolescents. Yet, e-cigarettes are pure nicotine delivery devices. And from the mass data that we presented, you see they will have the acetylation effect that we described, and other effects yet to be identified, potentially with the attendant risk for addiction to a variety of drugs and experiences. So, nicotine is nicotine. Doesn't matter if you get it from a combustible cigarette, from an e-cigarette, even though some e-cigarettes, in fact, don't even have nicotine. But that's another issue. So, I want to send my collaborators, Mai Chen Yu, Kristen Schaffer, and Carrie Case, Eric Kandel, etc. And the funding agency, but I guess the slide has disappeared, research support from the National Institute on Drug Abuse, the Truth Initiative, which used to be the Legacy Foundation, if anybody knows about this, and the Howard Hughes Medical Institute. Thank you.